Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, in the Q&A portion, and I'll ask our speakers your question. And the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Natalia Elizalde, PhD, Business Development Project Management Director, and Raquel Lopez de Maturana, PhD, Qualified Person Regulatory Director from Vive Biotech.
Good day, everybody. As I said, my name is Natalia Elizalde. I'm Chief Business Officer here at Vive Biotech. I'm very pleased to be here in this webinar together with my colleague, Raquel Lopez de Maturana, Qualified Person and Regulatory Affairs Director, who will speak after me. So to give you this webinar about the flexible and customized approach to lentiviral vector development and manufacture through a well-established platform. So firstly, just to understand who Vive Biotech is, we are a GMP CDMO, so contract development and manufacturing organization, fully specialized on lentiviral vectors, working on the development of lentiviral vectors from very early stages, having into account aspects such as cost effectiveness, regulatory compliance, or scalability, up to the manufacture for preclinical phases, manufacturing engineering runs, GMP batches for clinical trials, and also now being prepared up to commercial scales. So as a company, we were created in 2015, and since then, we have been working for 38 international customers. So our customers, they are mainly biotech and pharmaceutical companies, as well as some applied research centers, that they do have a pipeline of advanced therapies that encompass the use of lentiviral vectors. So since then, since 7 years ago, we have been learning about our partners' requirements, and that's why our priority is responding to the real demands, therefore giving answer to the real job to be done of our customers. And this is the reason why we thought this was the most appropriate title for our WebEx today, for our webinar today, with 3 key concepts: flexible, customized, and well-established platform. Also, in order to define the service we are offering, we are having into account the global market landscape. From a general point of view, as you know, there are more and more gene and gene-modified cell therapies under development. Indeed, this is more than 2,000 in July 2022. Moreover, the expectations in terms of the approved cell and gene therapy products, they are very good. FDA expects to approve between 10 to 20 by the year 2025. And also, well, the growth per year in terms of economical impact is expected to be more than 17 billion in 2035, considering billion dollars, US dollars. More specifically, thinking on lentiviral vectors, it's expected to reach more than $1,000 million, growing at an annual growth rate that is higher than 15%. And finally, considering the CAR-Ts, that, as you know, they are, some therapies whose most frequently selected gene transfer technology is lentiviral vectors, here the expected growth is very, very relevant. We are talking about, more than 30% annual growth rate. And the market size is so significant that we really think is something we need to have into account while giving the best response to our partners. So, which is our aim? Our aim is covering your requirements that we can define as the following ones. We should develop a gene transfer technology that is optimal for your gene of interest, also that is cost-effective. The process we are developing needs to be scalable to commercial. The response needs to be given in a timely manner. Of course, everything we are doing here at BioTech should be compliant in terms of regulatory related aspects. Offering an integral service, and something key for us is communication, so that we are working together as real partners. So we will cover one by one all of these points, both Raquel and myself, to show you how we are covering your requirements by covering this point. So firstly, starting with the gene transfer technology optimal for your gene of interest. Just a brief review about the lentiviral vectors properties. As you know, lentiviral vectors, they are RNA virus, large and envelope. They can be a pseudotype for a specific tropism. The most frequent one is VSPG, that is quite ubiquitous, but also different pseudotypes are being used to target in a more efficient manner the target cell. Lentiviral vectors deliver the genetic material directly into the DNA, and something very relevant to be mentioned is that they are transducing both dividing and non-dividing cells, among others immune cells that are one of the most frequent targets. The gene expression is long-term, and the transgenes that they can accommodate, they do have big size, up to 11 kb. And these lentiviral vectors can be used both for ex vivo therapies, the most common ones, such as CAR-Ts, TCRs, NK CARs, and so on. But also, importantly, there is a clear trend towards the use of lentiviral vectors for in vivo applications. Therefore, the vectors, they are being used as final products. We'll discuss about that later on. So considering the characteristics of the lentiviral vector itself, for us, something essential is being able to approach the lentiviral vector-related services in a flexible and customized manner. Therefore, we do have dedicated R&D departments as well as upstream and downstream optimization departments that are involved in different parts of the program, such as construct optimization, if needed, in order to really demonstrate or being sure to have a construct that is regulatory compliant and that is bringing the best titers as possible. Therefore, we are having into account aspects such as promoters interaction. Also, our R&D department and process development departments are working in new pseudotyping strategies to be able to reach the target cell in a more efficient manner. Cell line modification is being executed for some of our programs, for instance, in order to bring the lentiviral vector different properties, or also to do the production more efficient, or even to generate stable cell lines. Also, we are able and we are manufacturing GMP master and working cell banks, whose characterization is also well assumed by us, and finally releasing these GMP cell banks by our qualified person. USP optimization is also regularly executed here at BioTech, and for that we are adapting the transfection process by using different packaging plasmids, like adapting the transfection mix to reach the greater transfection efficiencies and so on. DSP optimization, this is also a key part of the process, so that we are reaching the higher recovery and the greater purity, as both aspects have an impact in terms of the functionality of the vector itself. And finally, we can use different fill and finish formats, both in vials and in bags, currently implementing also automatic capabilities. So the fact that we are following this flexible and customized approach makes key these two aspects. The fact that we are not only experts in manufacturing, but in virology, and also that we are a company fully specialized in lentiviral vectors. So more than 100 people now fully focused on this specific vector. Second aim, very important also to cover your requirements, develop cost-effective processes. The therapies that are being developed should be cost-effective in order to reach a greater number of patients. So that's why this is a priority, not only for BioTech as a company, but for the whole industry. From our point of view, cost effectiveness should not be only reached by scaling up, but also there are other manners. The objective is therefore obtaining more functional virus able to exert the final effect. And the final effect, in the case of an ex vivo therapy, is transducing the target cell, and in the case of an in vivo therapy, is the direct therapeutic effect. In order to obtain this more functional virus, we need to get greater titer of purer samples. Okay, so how to obtain this greater titer of pure samples? Well, for that is key having into account, again, the lentiviral vector characteristics considered for production. So the lentiviral vectors are continuously bad from the cells. They suit very well to perfusion culture and need to be processed and also stored in very well-controlled conditions in order to minimize losses. Also, these kind of vectors, they do have high sensitivity to different physicochemical parameters such as pH, temperature, shear forces, different chemical composition factors such as the salt content, among others. We are always having into account these lentiviral vectors characteristics considered for production, and indeed our R&D department and process development departments are continuously working on that, and we do have different publications on this specific matter, the key challenges and the new developments linked to lentiviral vectors. So considering all of that, we've developed our manufacturing platform. Indeed, we have manufactured more than 100 batches in reactors so far by following these manufacturing steps that are continuously fine-tuned and that you can see here. This is the upstream bird here. That is the longest one that takes approximately 3 weeks. And the DSP or purification bird, it's shorter. It takes around 2 days, but it's very important as well. So what we are doing is departing from a hectonine 3T cell bank. This is defrosted and expanded firstly in flasks and then in cell factories until day 12 approximately, when the reactor is seated. Once the reactor is seated, the growth continues until the adequate point when transient transfection takes place by using PEI as transfection reagent, the 3 packaging plasmids, and the transfer plasmid that is provided by our partner. Following, the harvest collection occurs during a process that takes approximately 3 days. And this crude wool harvest is the one that is purified by following this multi-step approach that encompasses clarification, endonuclease treatment to allow DNA removal, anion exchange chromatography to purify, sterile filtration, TFF to concentrate and exchange to the final selective matrix, and fill and finish in vials or in bags. In this manufacturing platform that we saw you, we are using a technology coming from Universals, that is a partner based in Brussels, that has some very interesting characteristics, such as the fact that this is a high-capacity technology. Therefore, the productivity per cell is much higher than by using other kind of prior technologies available in the market. Moreover, it allows us having a continuous harvest to maintain product yield. The exposure to harsh environments is limited, is minimized. So this is increasing the functional virus and also the performance. So the ratio between the P24, so total physical particles, and VCN, functional viral vector, able to transduce hectonine 3T cells and to integrate. The DSP is more straightforward. It's more straightforward because obviously the number of cells that are needed to reach the same titer, they are fewer. Therefore, the recovery, the purity, and the functional virus that we are obtaining is better. And also this technology is allowing having a low footprint, therefore smaller clean rooms that they impact on cost effectiveness of the process. This is a closed system, which is very convenient considering the sterile nature of this product. And also it allows us scaling up the process up to commercial phases. So this is indeed another aim we want to cover, to really cover your own requirements. That is having a process that is scalable to commercial phases. At the moment, at the Biotech, we are working in the manufacture of lentiviral vectors for different uses, both for ex vivo and in vivo therapies. For instance, approximately 65% they are being used for cancer immunotherapy, such as CAR-Ts, TCRs, NK CARs, among others. At 25% for rare, ultra-rare disease, mainly transducing hematopoietic stem cells, but not only. And now working for 5 programs in which the lentiviral vector is being used as final product, therefore in vivo applications. Here in this slide, you can see a list of the cells that are currently being transduced with the lentiviral vectors produced at Biotech. So TILs, tumor-infiltrating lymphocytes, NK, hematopoietic stem cells, mesenchymal stem cells, and iPSCs, among others. So the process we are developing is important to be scalable for both for ex vivo and in vivo applications, ex vivo applications that are transducing different kinds of cells. And this is something that is feasible by using these universe reactors that, as you can see, offer intermediate scales that are able to adapt very well to the different developmental phases. For instance, here you can see the smallest version of reactor available, Hydro, 2.4 square meter. Here we can use 1 reactor with corresponding DSP, 2 of these reactors in a row with corresponding purification steps, or 4 of these reactors in a row within its DSP to purify the harvest. In such a case, while using 4 hydro reactors, we are having 9.6 square meter batch sizes. These hydro reactors are optimal for developmental phases. Then, other kind of reactors available are carbo reactors, whose solid phase range from 10 square meter, 30 square meter, and also we can manufacture batches 60 square meter size. In such a case, we are using 2 of these 30 square meter reactors with a single DSP. Just for you to have an idea, for instance, 10 square meter is, regularly speaking, the selected batch size for early clinical trials for therapies such as CAR-Ts, TCRs, and so on. And 30 square meter is very adequate for late clinical trials for this kind of therapies. Thinking another kind of, for instance, hematopoietic stem cell transduction lentiviral vectors whose MOI, they are higher. 30 square meter is the selected batch size for early clinical trials, while 60 can be optimal for late clinical trials. Then we have this nitro reactor that can accommodate a solid phase from 200 square meter up to 600 square meter. And this nitro reactor is very good for late clinical trials up to commercial stages. Okay. Another point, another very important aim in order to cover your requirements is giving answer to your lentiviral vector manufacturing demands in a timely manner. And we are able to do it as well. Why? Because there is a rapid process transfer to reactor. The process to transfer from 2D testing to 3D in reactor is something very quick. There is a speed associated to this phase. We can say that there is a similar cell growth and population doubling time in 2D and in reactor within a similar productivity, and all of that make us the translation between 2D and 3D very straightforward. And the recoveries we are getting within these reactors harvest, they are higher than 30% up to 40%. Here you can see, for instance, a chronogram of a standard program. And I will not go in detail in every specific phase, but just for you to have an idea, we could accommodate a given program from scratch up to GMP in less than 1 year. So we are very competitive in terms of timelines. And something very important to be mentioned as well is that linked to the fact that we moved to a new facility last year with much greater spaces, currently we do have a good slots availability that can be given in a timely manner, which is key not only to accommodate new programs, but also, and even more importantly, to give answer to the existing additional requirements in a timely manner so that the program can progress without interruption. And now my colleague Raquel will continue with the next themes we are covering.
Thank you, Natalia. So I will take it from here. So next goal. Our next goal is to have a process and a product that is quality-based and regulatory compliant. To this aim, and regarding the structure of Vive, we have a quality unit that integrates the departments of quality assurance, quality control, and regulatory affairs. These three departments work in a completely coordinated way, directed by a single head, and obviously with a very close interaction with the rest of Vive's departments. Vive has projects from clients all over the world, the US, Europe, Asia, and this is why Vive's quality system is aligned with both FDA and EMA standards, and the quality unit has a global regulatory strategy approach. In this sense, we can give support in filling in the CMC sections of IMPDs and INDs, from just providing information to really authoring. And not just documentation for clinical trial approval. Vive had previous facilities at the Miqueletegui site here in San Sebastian in the north of Spain. But since we moved to a new building, the Tandem building, last year, we are now ready to produce lentiviral vectors up to commercial, as Natalia has mentioned. So accordingly, the quality unit is getting prepared to support the procedures and documentation requirements to take the lentiviral vectors to the market. The knowledge that the Vive team has comes from many different sources. First, experience. The experience that we have gathered along these years with more than 100 batches in bioreactor for many different clients with different characteristics and requirements, sometimes even hurdlers. More knowledge comes from our clients themselves. We have learned from them and with them. Experience with partners such as Cindia, but there are others as well. Knowledge from the inspections that we have had at the two sites, Miqueletegui and Tandem. And finally, the interactions that we have accumulated with many different regulatory agencies, the FDA, several European agencies, and also Asian ones. So going into inspections with a bit more detail, with respect to the former facilities at the Miqueletegui site, we had the first authorization as a pharmaceutical plant and GMP certificate in 2017. This certificate was renewed in 2020. And then we had another inspection in December 2021 for the authorization of the second plant, the Tandem building, and the corresponding GMP certificate. Our current authorization allows us to manufacture lentiviral vectors, both as active pharmaceutical ingredients and also as medicinal products. So in other words, lentiviral vectors that can be administered ex vivo or that can be suitable for in vivo therapies. With respect to audits from clients, as you can imagine, being a CDMO, we receive a lot of audits, and this is an increasing number of these sort of audits. In this slide, you can see a summary of audits from EU-based and US-based clients. And all in all, at the end of this year, Vive will have received 19 audits and inspections. Okay, Natalia has mentioned before that Vive covers all the phases of a project, from scratch to market. And here I would like to stress the involvement of the quality unit in all these phases of pharmaceutical development, and not only in GMP batch production. Starting with quality assurance and regulatory affairs, we ensure traceability and appropriate documentary support for next phase progression. We anticipate the requirements regarding suppliers, materials, equipment, facilities, services, and so on. And last but not least, the quality assurance unit prepares for and leads validations for the production process, analytics, and any other processes. Quality control. Similarly, it is the same department, same team that is in charge of performing the testing of all productions. And this team is completely aware of the advanced and the future needs of the project, so they can anticipate. For example, it is worth mentioning that the team applies the same technical protocol with the same acceptance criteria to non-GMP and GMP batches. And finally, it's important to know that Vive is authorized to perform release microbiological, biological, and physicochemical assays. Our analytical capabilities include analytical method development, tech transfer, and validation. In this slide, you can see some of the tests that we can offer. You will recognize some as the standard ones for lentiviral vector batch release. For example, mycoplasma testing, concentration by measuring the capsid P24 protein by ELISA, potency tests such as vector copy number quantification, which is the quantification of viral genomes integrated into the cell genome that has been infected, quantification of residual sequences from plasmids, from producer cells, endotoxins, the test for adventitious agents, sterility, quantification of residual DNA and proteins from producer cells, the test for identity, which is the sequencing of the viral RNA from LTR to LTR, the verification of the absence of replication-competent lentiviruses, both in the finished product and also end of production cells, and some other tests like endonuclease, appearance, pH, osmolality, full volume, and some others that are not listed here. Next aim is offering an integral service. We want to be that one-stop shop that will take care of your project, of your lentiviral vector production. Therefore, we engage very early in those first steps to get to know your plasmid, get to know your lentiviral vector production with that expertise in molecular biology and virology that we have mentioned. We can produce different types of batches in bioreactors, not only GMP batches, but also non-GMP batches, development, engineering, and GMP batches. We have an in-house QP for efficient batch certification. We offer regulatory, we can perform long-term stability studies as well as in-use or studies under stress conditions. Now, yes, regulatory support. We can perform we offer regulatory support for IND, IMPD submissions. And finally, and in line with that one-stop shop, we take control of all outsourced activities that we have to do for that lentiviral vector production, including analytical testing and plasmid manufacturing. So if we can summarize the last point, the take-home message would be, give us your gene of interest sequence, and we will produce you, we will provide you with a GMP batch of your lentiviral vector. And finally, and a very important point and very valued point by our customers is communication. And here we want to work with you as partners. So we aim for a fluent communication, a transparent communication, being honest and true, and keeping you updated. Regarding the business development and project management team, it is important to underline that it is the same team assigned to your project all along: negotiation, development, and execution for optimal coordination. And here we reach the final slide of this presentation, where we can see again all the points that we have covered today. Natalia and myself, we thank you for your attention and more than happy to answer your questions now.
As a reminder to our audience, you can go ahead and type in your questions to our speakers now. The question is, how do you think Vive Biotech is different? Why would you recommend contracting with services with Vive Biotech?
Okay, so yeah, I will answer this question. So well, there are different reasons why Vive Biotech is different, but probably the main one is that we are not only manufacturers, but also virology experts. Okay, so that's why we are working on programs, even from early stages, having into account aspects such as, of course, scalability, adapting the lentiviral vector to your own specific requirements, even having, well, some approaches that are differential, like new pseudotyping approaches or modification of saline. Of course, we are guaranteeing regulatory compliance. cost effectiveness by having increased titer as well as greater purity, and also, well, slots availability in a timely manner. So at the end of the day, our aim is giving answer to the specific requirements that we are continuously obtaining from our partners, from the market.
What are some specific issues related to the chromatographic purification of LVs?
Okay. So, well, as you know, the lentiviral vectors downstream or purification process is a process that must be very well optimized, linked to the intrinsic characteristics of the lentiviral vector themselves. That's why we do have a dedicated downstream optimization department that is continuously working on that. At the moment, we are getting a good recovery up to more than 30%, but still we know that we are using an anion exchange chromatography, and that some kind of optimizations could be made in order to get even a greater titer. So that's why we are working specifically on the chromatographic step to get a greater recovery and also a higher purity. This is one of our priority projects indeed. Now, if you want to complete something, Raquel?
No, that's fine. Yeah.
Okay, and what types of active yield do you see with your LVs following TFF?
Okay, so all along the DSP, we recover between 30-40% of active particles. I think it's a very good yield compared to what is known in the industry of lentiviral vectors. So 30 to 40% would be the answer to this question.
And what are the differences in terms of manufacturing and quality control assays, depending on whether the viruses are going to be administered into in vitro or in vivo?
Well, the main the first legal aspect is that you need a specific authorization for the production of lentiviral vectors that are going to be administered in vivo. We have 5 in vivo projects we are working in. The production process is basically the same, with some differences in some elements, like, for example, the quality of the raw and starting materials. And in terms of quality control, which is your specific question, for an in vivo lentiviral vector, there are some specific CQAs that you need to take into account, for example, aggregation. So we are working on all these additional parameters that you need to control for a lentiviral vector that is going to be administered in vivo.
Okay, well, it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Natalia and Raquel, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what are the main challenges in the manufacture of lentiviral vectors?
Okay, so, well, there are different challenges, because as mentioned during this webinar, the lentiviral vectors, they are, well, very somehow sensitive to physical, chemical parameters and so on. So that's why we are carefully working on all the different manufacturing steps, both during upstream and downstream. Also, one of the challenges is to really, well, being able to develop lentiviral vectors that are more and more directed towards the target cell, okay? And of course, something important nowadays is to have more and more automated process, as well as digitalized like manufacturing approaches. So all of them we are working well in order to be able to oversee or to overcome these challenges. Yeah.
From the quality point of view, first, for quality control, there is an analytical challenge. And to name a few, I would say to find tools for inline process monitoring. That's one challenge. Another challenge for lentiviral vector production and characterization is the test for biological activity. We still don't have a standardized assay for potency. we don't have a reference material, and that is a hurdle for comparison between laboratories. And then from the regulatory point of view, I would say that there is still not full harmonization on the submission procedures and quality requirements. And so the regulatory bodies and other professionals are working hard to correct this lack and to be prepared for the high rise in ATMPs that we have now and that we will have in the coming years.
Great. Well, thank you so much, Natalia and Raquel.
You're welcome.
And thank you to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.